although they know how to use the software, they have a little bit of trouble figuring out how to develop a new model. And that's because there are some differences in the thought pattern or epistemology of computational modeling versus kind of traditional biology or engineering practice. And so we want to kind of give you this experience in a guided way, and also give you the opportunity to improve your familiarity with CompuCell3D and to provide a venue for social learning. We found that peer-to-peer information transfer is really helpful. And so even if you're building a model on your own, I encourage you to discuss, especially during the kind of the road mapping part that we'll be working on primarily today, to discuss it with other members in one of the groups, because that kind of process of making your assumptions known and clear to others is one of the best ways to provide clarity in your own model building exercise. So we've got a picture here of kind of the lattice or the framework of a building, and that's really what I want you to think of in terms of what we're actually going to build over the next couple of days. We're going to follow up with more thematically focused hackathons, and those will have more direct support, and that will help help these models reach their production ready or research ready status, and or research grade status. And we're also going to keep the Slack open and provide technical support through that as well. So don't worry don't worry as much about having a fully functional, complete model by the end of the next 3 days. So we have 2 Google Drive folders that are available for you. So there's a viewable folder on Hackathon resources, and this is within the student materials folder. And just message either in Slack or here if you have trouble accessing that. And there will be a folder within there to upload your materials as you work. So upload slides as you and model components too, to facilitate sharing and viewing and those things. So what we're going to try to do today is we're going to try to identify possible new projects and then form teams around those projects. And then this is going to kind of be an iterative process. We're going to do this first teaser here with the 2 slides that you guys have all prepared, and then we're going to break out into groups and kind of discuss kind of a model conception formula and kind of work through that, and then we'll meet again, and I'll have the times are laid out later. We'll meet again and kind of refine that model, and we'll also have the opportunity to combine teams or switch teams at that point so that everyone can have the best educational experience. For those who So we strongly urge you to do that, even though it's a little bit slower and perhaps a little bit more boring to try to do the model definition aspect. If you insist that you just want to start coding today, we're going to have an option for working around this kind of COVID modeling that you've done, and that'll be in one of the breakout rooms and one of the Slack channels. But unless you actually work on viral infection, the process of transferring over from the examples you've seen to a new modeling domain is that's the thing that people have the most problem with, and getting that experience today, even if it's not on your exact system, I think is really valuable. So now I'm going to unmute and allow screen sharing, and I'll call people based on the order that they have. Yeah, I'll just call people and get them ready to present. So Rabia, are you ready?
Yes, I would be ready.
Okay, so I've stopped sharing, so I think you can share your screen at this point.
Okay, I'll try. Hello, everyone. So can you see my screen? And is it full screen now?
Yes.
Okay, perfect. So I'm from the University of Heidelberg. I'm a PhD student and I'm working on cells in 3D printed scaffolds. So we have a collaboration with a group that can do 3D printed scaffolds, as you can see here. And they can be any arbitrary structure at the micrometer scale. And the new thing now is that they're stimuli responsive. So with the help of light, they change their structure. And so previously we've done modeling of cells in scaffolds with surface methods and also a little bit with cellular POTS models. And what I would like to do during the hackathon is to use Multicell 3D to implement moving scaffolds. So as you can see the image on the left a little bit, the pillars should move during the simulation. And I think we have to think about how this can be implemented so that the cell probably is still attached to the scaffold as it moves. And then another idea that would be great, but I don't know how much time we have, would be to implement stress fibers in the cell for more accurate modeling. So those are my ideas.
Great.
Okay, you've already gotten all, I mean, you already have a CompuCell model, so you're pretty well along with that, it looks like. So that's wonderful.
Yeah.
So that would definitely be, I mean, having somebody who has experience is a great opportunity for people to nucleate some, a group around. That would be wonderful. Thank you.
Mm-hmm.
Yeah, thanks. So I probably should end my sharing. And then the next person, or do you have questions? I don't know.
So if there are questions, people can raise hands and then we can unmute to ask questions, or you can also type questions in the chat.
Okay, so if there's no questions, Mehdi, are you prepared to go now? Okay, we'll come back to him. Cyril, are you ready?
Yes, I am.
Okay, go ahead.
Okay, thanks. Okay, can you see my screen?
Yes.
Okay, good. So, hello everyone. I am a postdoc at the University of Liverpool in the UK. So I've been trained in physics, been doing particle physics before, and 1 year ago I started working on aging. So I'm interested in a very interesting aging process, which is a thymus involution. And so I have a people here. So we are already 3 people interested in this project. And if you are interested, just Slack me or email us. So just to give you a rough idea, I have a couple of slides with a few pictures of what we want to do and what is the problem. So the thymus here, the idea is to focus on mouse, okay? So it's a site of T-cell maturation. And as you can see, it is shrinking with age, and the total cell number is typically decreasing a lot in all vertebrates, actually. And it has a very complex dynamics and with different signaling pathway. So this is another view of a section of the thymus where the compartments, which are the cortex and the medulla, is becoming less and less clear with time, and the cells, the cell number is decreasing as well. So the idea is, can we build some models of this process and to learn about particular dynamics that could help us to understand why this happens, okay? Because this is a very important process for immune system in humans and invertebrates. And this evolution has been related to numerous diseases and cancer. So yes, you can see that there is a complex process of thymocytes, so progenitor T cells entering the thymus. They undergo proliferation, differentiation, apoptosis, until they get at the end and give rise to mature T cells. Okay. So interestingly, there are a lot of work on this topic, not really on modeling, but a lot of data. And one thing I found is that there is already a few ODE models, not for involution, but just for thymocyte development. So that would be a good starting point for us. So for example, during the week, I just took this model from 96 and I wrote it in Tellurium. It will not be, so you have this set of equations you can see. Now the idea would be to increase the complexity, adding things that people have discovered in the meantime, like interaction between the thymocyte and the epithelium, so the PEG cells, chemical signalings. And also here, this was a simple model where people were interested in the first seeding wave of the thymus. So you can see they have a carrying capacity here, which is a fixed parameter, which in our case is something that we want to predict, just to have this decline in numbers, okay? So we could start building this model of different models, ODE models, and then move to CompuCell 3D, as it has been shown during the week. And why a 3D model would be relevant is that because the epithelium and the shape of the thymus is very peculiar. You have these stellar epithelium cells which are doing a 3D mesh, and eventually a 3D model would be very interesting. And also there is a lot of motions inside it, a lot of dynamics. So these are the T cells moving in the thymus, you can see here. So there is a lot of things to do, and there is no model yet with CompuCell 3D. So the objective would be to start with something very simple this weekend, build maybe a quasi 2D model with an epithelium layer and some thymocyte layer moving into it, making the link with ODE models, and maybe later on adding more complexity. So thank you. If you have any question, you can always, I have much more material to show if some people are interested in Slack or later on. So you are very welcome to join us. Thank you.
Okay, thank you. Other people who want to present?
Yeah, let's have Mehdi next. Okay, I see 22 slides. Let's just make sure it's just a few minutes, okay?
You have to unmute. You should have permission to unmute.
Can you hear me?
Yes.
Actually, I am a PhD student at Lehigh University, and this is my second PhD, and my background is related to computational fluid dynamics. And I prepared this slide because, in rush, because I was in the middle of doing other projects, and in our lab, we just made a microvascular network, and we want to infuse the blood flow inside the microvascular network and see the growth of the cancer cells and the related phenomena. But our background in our lab is related to some CFD simulation, like squeezing nucleus cells, like fluid structure interactions and drug loading and this kind of stuff. That's why I do not have any experience regarding the biological system modeling. I followed your, I followed your presentations, and this is the modeling we can do in our lab. and I installed your package on Linux, and I found it very efficient. And I noticed that there are different capability in your software. That's why I have decided to use your model for modeling the fluid inside these microvasculature network. We have so many type of, this kind of microvasculature network, and we plan to see the growth of the cancer inside this network. For us, fluid is not important. Actually, we are, if we can, kind of omit the phenomena, fluid-related phenomena, and somehow we can focus on the cancer growth inside this complex network. It would be great for us. And also, I have read 2 papers of your group modeling of diabetes progress in retinal network, and also the growth of cancer inside the vascular network. That's why I felt that somehow I can go and use your software for this kind of modeling. I followed your presentation. I'm kind of familiar with Python and using your software on Linux, and I plan somehow and make a model for this problem. I'm hopeful to be able to do this. That's it.
Okay, I mean, I think people have done some simple models where you calculate an effective flow term using a Kirchhoff type solver, then you have the blood vessels grow or shrink in response to that. That's a possibility, although those Kirchhoff solvers take some work to implement since they're not intrinsic in CompuCell. Mm-hmm. The other thing you could do is that model of the cell squeezing through the channel. With CompuCell, the detail would be much less than in the simulation you show, because the mechanics are not as detailed. But one could replicate that model in CompuCell. You could take Gilberto's code for the crawling cell, put it in a channel, and then see how the mechanical properties of the cell change the movement. So that would also be possible.
That's awesome.
Let's see if there are other people who are interested in cancer modeling, and maybe we could get together a team to do a vascular tumor model.
Can Katie go next?
Sure. Can you guys see my screen?
Yes.
Okay. Hi, I'm Katie. I'm an undergraduate physics student at the University of Warwick. I'm just going into my final year of my integrated masters. I'm currently working on a summer research project jointly supervised by Dr. Marco Polin and Dr. Orkan Sawyer from the Physics and Life Sciences departments at my university. So our project is focusing on filamentous cyanobacteria. They're an important group of photosynthetic bacteria responsible for producing a large amount of the oxygen that we breathe. They comprise of various unicellular and multicellular species, ranging in size from filaments of just a few cells, like you can see in the images I've got here, up to large communities arranged in microscopic biofilms. The cells in these communities can be seen to functionally diversify into various metabolic conditions, aided by the simultaneous establishment of specific microenvironments. But our understanding of these structures and their dynamics remains quite limited. Cyanobacterial filaments exhibit gliding motility, so they kind of slide over surfaces, but we don't currently understand exactly how that happens. I have a little GIF here of some of the data from our lab provided by one of our PhD students. The current consensus is that this motility is a combination of directional slime secretion or surface waves that are propagating along the filaments as a result of distortion in the fibrils and the cell walls, or potentially even a combination of the two, we're not sure. The motion can also be seen to exhibit various instabilities like spiraling or buckling. You can see a couple of buckling instabilities in the GIF I've shown here, but not particularly closely. The genus we're focusing on also exhibits phototaxis. So in positive phototaxis, so they're seen to move towards light stimuli. This could have some really exciting applications if the behavior were to be quantified or controlled, even biological optical fibers, if we think really crazily. So there's a lot of data collected from one of the PhD students in our group, and there's a wealth of data in the literature that could be used to sort of compare with the model. The aim is to produce a working physical description of the filament dynamics to successfully describe the gliding motility of the cyanobacteria, as we currently don't understand it. Ideally, this would be in like a quasi 2D space, because that's where the data we currently have is. This would help to shed a light on the underlying physics at play, and particularly in the context of the phototaxis, because that's something we'd like to quantify. I've included a couple of references here of a couple of the existing models and some information about the gliding motility of cyanobacteria, just for some background information if anyone is interested.
Thank you very much. There were some papers on bacterial motility done a long time ago by Andreas Deutsch and his collaborators that used a POTS framework. It might be worth looking at those. You could create because you could create basically a string of beads with links and use those to model a filamentous bacterium. The other thing to think about, and maybe Andy, if you're on the call still, that might be a good, because of the mechanics of that problem, that actually might be a good problem for Mechanica. So it might be worth having you and Katie talk a little bit about whether Mechanica is in a position where it could do that simulation. It would be a little ironic if in the CompuCell course we passed you off to a new modeling methodology. But I think it's worth having a discussion about that, because Andy Saboji is developing a software tool called Mechanica, which is a next generation modeling framework. And it specifically is designed to model detailed biomechanics. And so potentially that could be something that would be of interest. So Andy, if you want to have a conversation with Katie early on to see if that's appropriate, that's fine. If there are any other people who are interested in bacterial phenomena, that would be great to discuss. I think it's a great project.
Okay.
This is Yi-Jung. I'm interrupting a little bit. I did some in the last 10 years ago. We did some myxobacteria simulations, kind of sort of a POTS model, but not quite. So I'll be happy to talk about that too.
Thank you, Yi. Yi hasn't been spoken so far, but she's been attending many of the meetings. She's a very, very experienced modeler, both with CompuCell and other techniques. She's on the faculty at Georgia State University in Atlanta.
Okay, if we could have Ajay next.
Hi, I'll try to share my screen. Whoops. How does this work? Oh, here we go. Okay. Can you see my screen?
Yes.
Okay. So my lab studies this beautiful structure, the lateral line primordium. It migrates in the zebrafish embryo from near the ear to the tip of the tail. And as it goes, a lot of interesting things happen. The cells reorganize inside to form these little epithelial rosettes. And in the center of these epithelial rosettes, a cell gets specified as a sensory hair cell progenitor, which is going to form the sensory hair cells of this sensory organ that's being depicted. And we know from a lot of work that the Wnt, Notch, and FGF signaling pathways play an important role in coordinating cell fate specification, movement, morphogenesis, all kinds of things. We have a basic network that we know operates. We have Wnt signaling promoting itself locally. It produces these FGF factors that migrate at a distance to set up FGF signaling at the far end, where it helps set up a neuromast. I'm not going through the details because it'll take me too long, but essentially the pattern of FGF signaling initiates a pattern of this 801 expression, which helps cells get specified as sensory hair cell progenitors. Notch signaling helps to restrict that to a central cell. That cell becomes a source of delta, which activating cells, which helps to make those neighboring cells support cells, but it also does something very interesting. It changes their cell behavior and makes them form an epithelial rosette by making them interact with each other in a strong way. The FGF signaling also inhibits the Wnt signaling, and this eventually pushes the Wnt system, which was initially very broad, to a smaller domain that allows another one of these neuromasts to form. And that's how this thing works. This process continues as more and more are formed. It turns out that the FGFs that are produced in the front not only help to make these rosettes, but it also serves as a migratory cue for these follower cells. And so the whole system keeps moving. The Wnt slowly shrinks, and the trailing cells eventually lose their ability to migrate, and they get deposited in this manner. I'm going over this like super fast, but I'm just trying to keep it short. So this is how the thing happens. For some years, we tried to understand how this simple network might create these little spots of FGF activity, in the context of which notch signaling plays a role in help specify central cells as progenitors. I already have I have no experience with Python or CompuCell, so actually I had quite a hard time keeping up. I've worked with NetLogo, which is a very much simpler language, and we had made this model to try to visualize how local activation of Wnt coupled with long-range inhibition of Wnt via FGF sets up this reaction diffusion system that can pattern the periodic formation of these little FGF signaling centers, which seed the formation of these rosettes. So that's the sort of level of modeling I had been familiar with, but I'm trying to now expand on this to answer many, many questions that could not be done because of the limitations of NetLogo. So I have to try to help me get more familiar with CompuCell, because I need to catch up quite a lot. I've tried to define 3 different small projects for me to do. So the first is, I mentioned earlier on that the central delta expressing cell activates notch, and this helps to change the morphology of the cells. So one of the things I wanted to do was simply start with modifying the delta notch program to make cell-cell adhesion proportional to the level of notch activation in neighboring cells, and see how that changes the morphology of cells. That should be a simple starter project for me. The the second project was to try to build a model just representing a rectangular block of cells that represents the primordium, and try to specify cell-cell adhesion between the different types of cells that would help me see if whether modifying adhesion can naturally lead to the cells adopting the shapes that they normally have in the primordium. I skipped over something that, oops. I skipped all over. So normally we see the primordium moving like this laterally under the skin, but it's actually moving. So this is a different view looking from under the skin, and this is a schematic that corresponds to that view. So you have actually the skin on the top, you have these sort of loosely organized cells in the front, and the trailing cells are reorganized into these rosettes. So I wanted to try to make a model of just the cells representing that and see how changes in adhesion between the cells might allow the cells to approximate this. And then the third project that I thought I would do is just simply try to make a model of this regulatory network, which I've already done in NetLogo previously, but I want to try to learn to do it in CompuCell. So those are my ideas.
Okay, well, you're quite far along if you have the NetLogo model. And NetLogo is a wonderful language. You can do an awful lot with it in a very compact way. I find it a very appealing way to do things. One thing to think about possibly in this is that there really are three separate things going on, although they're all happening together in the biology. One of which is collective migration of the lateral line cells.
Right.
One of which is the rosette formation, and one of which is the delta notch signaling.
Yes.
And it might be a good idea to start out by breaking those apart and trying to model them independently.
Correct. That's why I defined
Put
3
it
different
together.
projects to do exactly that sort of a thing.
But I don't know if Julio is going to be available to help with his delta notch signaling. Other people in the group also can help with that.
Yeah.
The collective motility is something, wound healing and collective motility. I think, Giuliano, you've done some work on that. There's some nice papers, not so much on lateral line migration, but there's some nice simulations using other methods by Paul Kulesa looking at neural crest cell migration. Those could almost certainly be replicated in CompuCell to do the collective migration. And Priyam, who lectured at the end of yesterday, again, I don't know what her availability is going to be today.
Yes.
She has worked on rosette formation.
Yes. No, I was very excited to hear her talk yesterday.
So I hope she can help us out. She's traveling today, so I don't know what her availability is. But definitely those are all three things that would be something that could be modeled. And we've talked for years about trying to do lateral line modeling and never had a specialist like you who was going to push it. So that would be great.
Oh, I'm looking forward to this.
Thank you.
Then we'll have Josh next.
Yep. Okay. Can you see my screen?
Yes.
Okay, good. Well, I want to thank the organizers of the hackathon. This has been a really educational week, really exciting to learn about the capabilities of CompuCell 3D, and I'm happy to share the story of why I came to the workshop. My name is Josh Laxina. I am a clinical fellow in infectious diseases at the National Institutes of Health in the US. And I work in a lab that studies, we like to say, the most deadly animal on Earth. And this is true. The deadliest animal on Earth, as some of you may know, is the mosquito. It's killed, at least in 2015, over 800,000 people, which is more deaths than any other animal, including humans, human-on-human deaths of any other species. And while it's appreciated that mosquitoes and different species of mosquitoes transmit a lot of deadly infections, what has been underappreciated is the role of the bite of these insect or arthropod vectors and the role of that initial interaction between the bite of the vector and the skin and how that influences the pathogenesis of infection. So I wanted to share two brief stories to illustrate just how dramatic this effect is and how important it is that we understand it. The first comes from a mouse model of viral infection with an arbovirus, a virus that's transmitted by a mosquito. And what the investigators tested was what happened if they delivered this virus via a needle injection, which is what nearly everybody does, a syringe needle, versus delivering the virus along with the bite of a mosquito. And first, what the investigators found is that the ability of the virus to enter the central nervous system, a privileged site, the brain, was dramatically enhanced by over 6 orders of magnitude simply by the presence of the mosquito bite, as opposed to the virus delivered by the needle alone. So this is a dramatic effect. So not only does it enhance the dissemination of the virus, but it also converts a strain of this virus from one that doesn't cause any death to one that is lethal in 50% of animals. So this is all due to the effect. This increased death is due solely to the presence of a mosquito bite. So something really important is happening here. And this is a theme that's repeated over many different vector species, over many different kinds of pathogens, ranging from viruses all the way up to protozoa, even up to parasitic worms. And it's this idea that the bite of these insect vectors or arthropod vectors exacerbates the dissemination of these pathogens and even enhances disease, converting something that doesn't cause any death into one that kills half the animals. Now, the flip side of this interaction comes from these experiments from our own group, where we looked at the parasitic infection Leishmania, which is a protozoan parasite, and it's transmitted by the bites of sandflies. And we tested the ability of sandflies to transmit Leishmania in a non-human primate model to these rhesus macaques. And what you see is that when this is the form of the parasite that infects and causes disease in the skin, when the Leishmania parasite is delivered by the sandfly, it causes this terrible, ugly-looking lesion in the skin that ulcerates and is chock full of the Leishmania parasites that eventually infect macrophages. But what we've found over 2 decades now is this fascinating theme in the field, which is that there are specific proteins in the saliva of these vectors where if you immunize animals with that saliva protein, you can actually protect them against disease partially. So here we used a sandfly saliva protein vaccine candidate, vaccinated these non-human primates, and you see no ulceration and very mild lesions. And if you take samples of that skin and count the number of parasites in them, you see that there is a 2 order of magnitude decrease in the amount of parasites if you just vaccinate with one of these salivary proteins. So the flip side of this immune vector bite interaction in the skin with the pathogens is that we could leverage that to our advantage, and that vaccination with some of these proteins in the saliva can actually protect against these infections. And this theme has become so well established across different animal models that 2 months ago, our group participated in the first in human phase 1 clinical trial of this approach and demonstrated the safety of using mosquito salivary peptides as a vaccine candidate. So there is a lot of a lot of exciting immunologic observations that illustrate the important biology, the importance of the immune response to the vector bite in the skin, and that how that influences downstream infection. And so my goal through learning CompuCell 3D and what I'd like to pursue in the hackathon is to be able to look at this complex network of interactions for when the vector bites and instills saliva into the skin, and how that initiates an immune response that can lead either to pathogenesis or, in the case of vaccination, to protection. And so the broad questions I'd like to address with the model are: what are the immune parameters within the skin immune response to the vector bite that exacerbate the severity of infection, like we saw with the arbovirus model and the mouse data, the virus that got into the brain? And can we find, like TJ demonstrated with the COVID-19 model, are there a range of parameters where we can determine what leads to deterministic versus stochastic outcomes of infection? As you might imagine, when you look at human patients who have Leishmania or dengue, and true for COVID-19 too, not vector transmitted, but you see this range from asymptomatic infection all the way to infections that cause severe disease and death. And what are the parameters that dictate such a wide variability? And to what extent can we identify the parameters space where that is stochastic as opposed to an outcome that's determined by host immune makeup or genetics? And then what are the parameters that are going to determine the efficacy of these salivary protein vaccines? So these are very broad strokes. I understand I think that my approach to this modeling, I'm really going to have to spend time working with the team and with the organizers about kind of distilling this down to the core elements and making this much more focused in terms of what we want the model to investigate. But that is what I would like to present for my project. And as a brief aside, I mentioned this in the Slack channel, but I wanted to raise it because I want to hear input and get ideas from people, is that my broader interest within systems vaccinology is, if you look at systems biology, I would say within systems immunology, the majority of the focus in the field has really been on this high dimensional, multi-omics, single cell omics across multiple scales, looking at all different kinds of parameters. A lot of machine learning approaches really looking more at signatures. But I think that's something that's been missing in the field, is to take the multi-omic data and translate it into quantitative and mathematical models like you all have been doing with CompuCell 3D. So I would love to hear from all of you and learn strategies so that we can do a better job of this, of taking that multi-omics data and really trying to better understand how the elements of it interact with each other in a quantitative way through modeling. So this is me. Again, I work in the Valenzuela lab at NIH in Maryland in the United States, and I would welcome input, additional members of the team, and I want to thank you for this opportunity.
Great. Thanks.
Okay.
I think that helps people. Go ahead.
Thank you. It's a fascinating problem. I think it's as you say, the key thing would be to think of what we could do productively to narrow it down over 3 days, so that we could come up with something that would be a good start for addressing it. I think this idea of trying to understand some of the immune response is wonderful. Are there any models, ODE models or other kinds of models of any of these components of these systems that you're aware of?
The 2 models of the skin that I've seen are, there was work that's now 10 years old by Frank Nestle, looking at skin inflammation in the context of psoriasis. Interestingly, rheumatology and kind of skin autoimmune disease, there's been some mechanistic modeling work. So, and I'll admit I haven't looked at that work in detail. This was just a cursory kind of Google Scholar search so far. So Frank Nestle from 2010, and then Reiko Tanaka's group at Imperial College London. She has developed this interesting model of atopic dermatitis and explored some of the parameter space, even looked at some of the, she's built in switches as part of her ODE model. I had tried a little bit last week at Tellurium to try to translate that into the Tellurium model. I don't think any of it has been cellularized or really put into space in a meaningful way. So, but definitely within this specific problem of vector bites and the inflammation and immunity to vector bites, Dr. Google Scholar says there are no modeling studies of this. I think the broader theme of mucosal immunology, and why I think it might appeal to people beyond this very focused problem. I know that there's been work that some of you probably know about from the group at Virginia Tech on modeling GI tract immunity to Helicobacter pylori. My brief glance at their models, I saw it had a lot of parameters. I haven't looked at it in detail. And those are the main ones that come to mind.
Okay, thank you. Anyone else have? Thank you, great. What I'm going to do, and if you hear me typing in the background, I appreciate your patience, is I'm going to, once we've gone through everybody's suggestions, I'll do a quick poll where I ask people whether, list all the suggestions so far, ask people if they have a preliminary feeling about whether they'd like to join one of these teams that's been proposed. And then we could look at that and see if there's some ability to rebalance or assign things, okay? So I've been trying to type up a little bit about what I'm hearing to make sure that we capture all of the interest. And if there's people who have interests or suggestions for projects that are not being presented, that's fine. Send them to us through the chat or the Slack, and we'll add those to the polling list and try to come up with a set of teams, okay?
So I have 3 more on my list now. So if we can have Evan go.
I'm happy to. Thank you, Bobby. Hopefully everyone can see me and hear me. I'll share my screen. Okay, does everyone see a PowerPoint presentation?
Yes.
Perfect. Okay, my name's Evan. I'm a third-year PhD student at NC State University in the biomathematics program. For the most part, recently I've been interested in continuum mechanics modeling. So CompuCell is certainly a new foray into a different type of modeling. I don't have experience with cellular pots, very limited experience in Python, but it's been fascinating so far. The problem I'm interested, I've already stripped it down a lot, and I'll try and briefly describe the sort of sandbox model I'd like to try and figure out over the course of the next 3 days. And that's tube remodeling. So very, very common phenomenon in early development. And tube remodeling, the tube itself, the topology will remain the same. We're not talking about growth in this context yet. We're just simply talking about the remodeling or the changing of the neighbors of cells. So there are some topological changes that can happen. The most common one that gets brought up a lot are these sort of type 1 to type 2 configuration changes where you have cell neighbors switching. So in the bottom right, you have cells 1 and 3 are not neighbors, cells 2 and 4 are neighbors. And due to myosin contraction, they will form this 4-point meeting spot, and then we'll switch, and 1 and 3 are neighbors and 2 and 4 are not neighbors. So there's a sort of local phenomenon that's occurring, and it's involved in cell intercalation and these sorts of things. But I'm interested in tubes. So like I said, I was doing continuum modeling. So this is a surface evolver model, and it's just a toy model to look at how changes in surface tension, this apical constriction phenomenon that Priyam was talking about yesterday, can describe changes in cell shape and the geometry of the overall tube. But I want to extend it a little bit to a case where we have multiple layers of cells. So that second picture, there's the lumen of a tube on the inside, and you've got the basal membrane on the outside. And so I'm curious how changing things like cell-cell adherence and cell membrane surface tension, and maybe even possibly adding some element of polarization to a CompuCell model, can give us an outcome like on the far right, where we finally get to a ring. So in 2D, it would look like a flat ring, but in 3D, there could be multiple outcomes depending on, my guess is density parameters based on some of the papers I've read. So the cells in 3D could be stacked differently depending on the density of the cells in the tube and how they adhere to each other. So you could have something like this bamboo configuration where they're all stacked neatly on top of each other, or you could have this sort of staircase topology where they form, I guess, what looks like a staircase. So anyway, this is a relatively simple toy model. My goal is just to pare down this problem into something that's manageable. So if anyone's interested, let me know, and hopefully we can get something interesting out of it and try to extend things. Thank you.
Okay, thank you. That has some, of course, some overlap with the lateral line reorganization problem. Giulio didn't have a chance yesterday to show his work on convergent extension. I don't know if you've talked to Giulio already, given that you're both at the same institution. Sharon is an old friend, of course, of mine. And I think, I mean, to begin with, one could try to replicate some of those existing results of Sharon's and see if well, not just Sharon's, but the existing results you showed in 2D, and see if you could get that to work. The links function in CompuCell allows you to do convergent extension, and Giulio's published convergent extension model using CompuCell.
That is the hope. Yeah, thank you. Giulio is on my committee, so we've talked about it in briefly.
Okay, so you've been talking about it, but you haven't implemented it yet.
That's correct.
Okay, great. So that's good. As long as the domain expert is somebody you're in contact with, that's great. Okay, thank you.
And then if we could have Romain next.
Yes. So yes. Can you see my screen? Okay. Oops. Yeah, so my project is about ovarian folliculogenesis. So it's the process that makes the germ cell maturate and grows that leads eventually to the ovulations. So it's a very complicated system, as many in biology. This one is intricately related to the central nervous system, which coordinates signals, neurohormonal signals that diffuse through the whole body, so from the brain to the gonads. And inside the gonad, you have a bunch of guys who are the ovarian follicles. And these guys are composed of the one germ cell in the middle and surrounded by a bunch of somatic cells. So it's a mini tissue in a sense. And this follicle, so we'll start with very few somatic cells. And all along the life of this ovarian follicle, it will grow from micrometers to millimeters, and from around 10 cells to 10 millions of somatic cells. And the germ cell will grow together. And so this process is obviously controlled by lots of neurohormones and interaction in between the population of follicle through the reproductive life. And in mammals at least, and in humans in particular, this process of growth of ovarian follicle is linked to many fertility issues, and also the main target of treatment, of fertility treatment. So my ultimate goal is to model the growth of one ovarian follicle in interactions through ODE or PDE models with the follicle population and the neurohormonal signals. So that's probably not a reachable goal for a weekend, but that's my track. And here I'm drawing what could do the model for the initial steps. So initially we have the germ cells surrounded by a bunch of somatic cells in a ring around the germ cells. And when it's initiated, so it leaves its quiescent states, the cell starts to proliferate. And so first a single layer with hundreds of cells, and it becomes a multi-layer tissue, and reach probably some states that might be hard to compute numerically, at least in a laptop, with a million or 10 of millions of somatic cells. So the first steps of growth, I think, is quite similar to tumor growth, with the except that in the middle there is germ cells that is also growing. But there is also an interesting steps in the bottom, which is a cavity formations called the antrom in our field. So it's a liquid filled cavity that appears in quite conserved diameter in all mammalian species, so around 200 micrometers. You have this cavity that appears and plays very interesting role to support the growth and the proliferation of somatic cells. And currently, so there's a bunch of hypotheses to explain how and why this cavity appears, but not decisive hypothesis. So one of the interesting outcome of this model would be to propose one or several mechanisms that could lead to this cavity formation. Yes, so that's what my scientific question here. And a few words on the data that we can get. So there's very few data, and in particular, no kinetic data in this kind of object, which are very difficult to measure in vivo. But what we can have is a morphological measurement, so snapshot in a given individuals, either in humans or in sheep or in rats. And so you typically have this kind of relation with the number of somatic cells and the diameter of the whole tissue, of the whole follicle, which can give you the size of the germ cells, the size of the cavity, and so on. So this kind of relation is definitely to be followed by any mathematical model. And then you also have count population data, the number of total follicles within an ovary or within 2 ovaries. Okay, so that's my project.
I guess for that one, Jim Sluka has talked to some people about ovarian development. And Joshua, who is on another call right at the moment, but will be joining us soon, has done a little bit of modeling on cavity formation.
Cool.
He's been modeling not so much this stage of development, but he's been trying to model blastula formation and the formation of the cavity there.
I've been looking some work on that. Okay.
And so he's thought about some of those problems a bit. And those are both 2D and 3D models. And 2D is much faster. 3D, of course, a lot of things don't work so well in 2D because the connectivity is different in 2D from 3D. As you say, getting to millions of cells is probably a stretch, but at least the earlier stages you could probably do. And sometimes if you could get away with a model where one cell represents 10 cells or 100 cells, and it still has the right basic properties. So I think it's quite an interesting problem. Defining the processes and interactions is going to be challenging, but it's certainly the kind of model that could work well in CompuCell. It's another model where the things that Andy is doing with his next generation environment could also work. So it would be interesting to try that as well. But I think that's a nice problem in terms of the level of complexity mechanically, and potentially CompuCell could answer some nice problems there. So that's great. Okay. So one thing that was interesting to me is we thought that this year everybody would be interested in viral infection and COVID, which is why we retooled everything to talk about viral infection modeling. And so far, there's only been one person who was interested in viral infection. So if we wound up using a demonstration problem that wasn't the most ideal for you, I appreciate your patience. These classic developmental problems are ones that CompuCell has been used for historically a lot, and they definitely are things that CompuCell is very good at. Okay, are there other presentations to do?
Yeah, Divyaj is the last one. If anyone else wants to go, they should chat me as well.
Hello. Yes, I hope the screen is visible and I'm audible. Is it?
Yes. Mm-hmm.
Yeah, thanks. So, hi, I'm Divyoj, an undergrad in Institute of Science, India. So, I have a very naive project actually to say. So, yesterday we talked about notch signaling, and we saw how basic equations and interactions among cells can lead to different behaviors. So here I want to link another network and a phenomena with the notch signaling, which has been worked out in recent times, but through a different approach. So there is this phenomena of epithelial to mesenchymal transition, which is associated with cancer growth and development biology as well. So basically what happens, say there is a cancer tumor, tumor, and then cells from one part of the body transition into mesenchymal cells, enter the blood channel, move to different part of the body, and then again go back to epithelial cells, and then start going there. So this transition, how a epithelial cells transits into a mesenchymal cells and then again transits back to epithelial, is not very well studied. As in, it's being studied a lot, but it's not very well understood as of now. So there is this model of these four key players, and then people have worked out, including my PI, who has worked out very detailed, in which they show that the system, this network has 3 steady states, and it is as in different parameter regimes. It has 3 steady states. One is a mesenchymal state, there is one epithelial state, and there is one hybrid state as well, which is somewhere between epithelial and mesenchymal. Then they worked along with the notch signaling, and so there is this. So these two networks are very well interconnected, as you can see. So there are the notch and the notch and delta genes, which we saw yesterday. So those interactions. But there's another player in the notch signaling as well, which is jagged. So, and jagged is also jagged is like a competitor of delta. So when these two cells, when these two ligands interact, the cells bind to each other, and this is how the signaling happens. So inside there is another network of EMP, and these two networks are very well connected. And people have they have done done simulations on Python in which they found that there are 2 states when you consider a whole cell. There are 2 states of a sender in which there is high notch, and receiver in which there is low notch. And in different scenarios, so yesterday, as you saw, there was salt and pepper behavior on the sheet, but you also see you also see a hybrid state in which a cell can achieve a sender or receiver state. So this is something related to EMT, what we can see. So then they moved on to do a 2D lattice. So here the cells are all hexagonal, in a hexagonal lattice. So each hexagon is a cell. And then since in the network there are terms of external notch and so notch levels of the surrounding cells. So they see that when the jagged signaling is dominated, there are clusters of mesenchymal and hybrid cells formed, which is not the case with notch signaling. So when the notch, which is not the case with with the delta signaling. So when the delta signaling is dominant, you only see salt and pepper behavior. But when jagged is dominant, you see clusters of mesenchymal and hybrid cells form. So we can, I think, so this is on a hexagonal lattice. So, but we can try doing this in using the cellular plot model and see whether the behavior holds true now as well. So we can see whether the jagged dominated signaling gives you the same clusters, which we are seeing in this simple model there, where we had just considered one hexagon as a cell. But here we can have much more dynamics and stuff. And what are the effects of other parameters? So this, even this behavior is not very well studied. We can look into that. Also, we can link the notch, the levels of notch with the adhesion strength and everything. So yes, this is what I was. So since it's a very simple model, I think we can do it by 3 days. That's my hope. Yeah, I guess I'm done.
Okay, that's great. I mean, when you have an existing cellular model like that, replicating that one in CompuCell would be a great place to start. And then, as you say, you can lift some of the simplifications. You can make the cells motile. You could have the cells vary in their properties and so on. So I think that is a practical model to try to replicate in a few days.
I have a question or a comment. This is Ajay.
Please go ahead.
I was wondering whether you had considered that, unlike the delta notch system, where they are usually mutually inhibitory, that that the cells either have high delta or high notch, that with the jagged system, it's actually self-reinforcing. So notch actually increases the level of jagged, at least in some situations, and that may make the difference between whether you get a salt and pepper pattern or whether you get clusters formed. So I was just curious.
Yes, true. As in that is, yeah, true. That is linked with the EMT circuit itself. So that is part of the reason why you get clusters.
Right.
So because the EMT network is such a way that it that notch in turn inhibits delta and activates jagged. So that is part of the reason why you get clusters, which in its own glory is beautiful.
Okay, great. All right. Anybody else have thank you. Anybody else have something they want to present? Bobby, do you have any?
No, I think that's everyone who's reached out to me.
Okay. Let me set up a quick, now that I have the list, let me set up a quick poll and just get a sense of what people, the people who didn't speak, what their interests are, who might want to join a group that's existing. Give me about 2 minutes to do that. Is there anything else you want to go over now, Bobby, before, if people want?
I can pop up the schedule while you're doing that.
Great.
So how do I do this? So Divya, can you stop your screen share?
Yeah, sure, just a minute.
Okay. So obviously we're running just a little bit behind on this, but that's fine. After this poll, Dr. Glazier is going to show us, do a quick walkthrough of the worksheet that we have for you on model definition. And I think at that point, we'll break out into Slack channels and breakout rooms to kind of go through that worksheet in detail. For the most part, you guys will work on it together within your groups, and we'll have different CC3D faculty members pop in at different times to see where they can help, and they may be particularly helpful in referring you to existing models to draw from. Then we'll have a quick check-in at 3:30, and that'll be in the main Zoom channel. And this will be an opportunity for us to reshuffle teams as needed, and also make sure everyone is developing a good a roadmap for their plans for this week, and then do another work session, and we'll have a final check-in 5:30 to 6 p.m. We'll keep that general structure of a morning, afternoon, and kind of evening check-in throughout the weekend. And if you can't make one of those, it's fine. Just let us know so we're not waiting for you. We understand, obviously, with it being geographically distributed and being the weekend, there may be, you know, uneven availability. So just kind of work that out within your groups. We'll develop a Slack. We'll have a new Slack channel for each team, and then we'll keep this Zoom open, or you can just, you know, you can use your own Zooms for working in between that time. But at least for today, let's try to use the breakout room so that to make it easier for faculty to, for the CC3D faculty or grad students to drop in on you. So this is that day 2 schedule that I mentioned. And then the day 3 schedule that I mentioned.
Okay, so I'm going to put up, I think I have, so the list of potential projects that I had were tissue engineering scaffolds, thymus model, vascular tumor modeling, cyanobacterial motility, lateral line development, mucosal immunology, tubary modeling and convergent extension, ovarian folliculogenesis, epithelial to mesenchymal transition signaling. And is that everything? Did I miss anybody? Okay, if that's, we can always do this a second time. So let me launch this poll and get a preliminary set of ideas about who finds what interesting. And then somebody says, please allow multiple selections if possible. Unfortunately, I don't believe I can do that, but I can. So why don't you indicate the one that your first choice, and then we could do it again and get a set of second choices. How about that? Okay. Even if you're the person who suggested the project, please click your own box so that we can have get a estimate of how many people are going to be in each section. And I guess a few people still haven't voted. Do people need more time? You can always change your mind, so it's not that this is a definitive allocation of resources. But it looks like there are at least 2 people in each category, which is good. There are a number of people. 4 people have indicated they'd like to suggest an additional project. If you're one of those 4 people, please send a. I'm sorry about number 2. I should have had a none of the above option. My mistake. I didn't realize it forced you to answer all of the questions.
Okay, so most of those 17 are actually not.
Okay. So I guess then, so is there anybody? My mistake learning how Zoom polls work. Is there anybody who actually wants help selecting a project? Why don't you just type into the chat if you are? Okay. So please, if you want help selecting a project, please text Bobby and with your name, and we'll go into, we can do that.
Okay. Yeah, and I have your application material, so I can help sort too.
And is there anybody? Let me get through it. So there, I see a couple of people. 1, 2, 2 people said they did. 3 people want help. Is there anybody? Is there anybody who would like to suggest an additional project? Not, you can always do it later, but it just now is a good time. So nobody seems to really want to suggest an additional project. I had as an option if people want to work on some of the COVID-related models from earlier in the class, but maybe does anybody strongly want to do that? No. Okay, so it looks like we have 8 projects. 8 projects so far that have and there's a group and how many? So we have 3 or 4 people who are unassigned.
Wait a second. I thought there were 9 presenters.
I'm sorry, I said 9. I said 8. I should have said 9. Thank you. So there were nine possible projects, and it looks like there are at least a couple of people in each one, which is good. So let us, and there, let's see, how many people are missing here? 2 people said none of the above. So there are 2 people who need to help making up their minds. And my suggestion would be that those people join a project which has fewer. Does anybody still need time to fill out the poll? I'll give you just 1 more minute. I'll share the poll results here. So my suggestion would be that people who are undecided join one of the groups that has fewer. Can everyone see the polling results? I would suggest that people who are undecided try to join one of the groups that has fewer people in it. It's also possible that some of the smaller groups might want to consider joining forces. So, for example, the epithelial to mesenchymal transition model, the tube remodeling and convergent extension, and the lateral line development model all have somewhat similar or overlapping components. And so there might be a way of combining your ideas together to think about putting those things together, because you'll have to develop some shared modeling infrastructure for all of them. But I think in general, if we have a list of the people who want advice, we can go into a breakout room and discuss that. And I think, but I think we've got a good distribution. There were 9 people who expressed interest in vascular tumor modeling. That's fine, and having a big group is fine, but I might encourage some of you to think about possibly, depending on how definitive your interest is, maybe think about maybe joining one of the other components. A lot of the issues that will come up in the vascular tumor model would also come up, for example, in the tissue engineering scaffold model. Some of them would come up in the thymus model or the folliculogenesis model or these tube development models. And so that's something to think about. All right. But I think we have a place we can get started with. I think that's all possible. So the people who are unassigned, we're only a couple. Is there anything we need to go over? Any urgent questions before we get started?
I have actually a question.
Go ahead.
It's kind of an embarrassing question in that I really had a hard time in following the course. And so in some sense, the exercises I had designed as part of the broader lateral line project was starting from a very simple modification of the delta notch thing to slightly more complicated, with the purpose of helping me get on, learn to do the programming better than I've been able to so far. So I'm not sure exactly how this will work if we all do it, a large number of people do it together.
Well, you could, depending on how the group works out. I think there were, the last presentation, let's see, I'm sorry, I can't see who's talking. Okay.
Actually, I got an email from Yi saying she would like to work with me.
Right.
So actually, in some sense, that certainly is a good idea, because she could help me, be like a tutor to me a little bit.
But also there was, the last presentation was also on essentially on delta notch-like signaling.
Yeah, he had a similar, actually, it's very similar to the first exercise that I was thinking. Yes.
So that's what I was saying. I think some of these could be potentially combined. Just because, you don't, if you're in the same breakout room, it doesn't mean that everyone has to work on the same thing at the same time.
Yeah.
I'm more concerned because there aren't that many instructors to circulate.
Right.
I want to make sure that everyone gets some attention. And if you're there sitting on your own struggling, that may be all right, but it could be frustrating too. And so I was wanting to avoid was a situation where we had 24 different single person projects.
No, I appreciate that.
And so to make sure that everyone was able to work collaboratively, because one thing we've learned definitively over the years is that nobody comes to this kind of modeling knowing all the pieces. And so you need a variety of expertise, biological expertise, software expertise, mathematical expertise, physics expertise. And so if we can team people together where one person maybe is stronger in programming or has used CompuCell before, and one person has really good biological insight, those kind of teams tend to be productive.
Sounds good.
And so, but absolutely, this is all for you. We have no agenda except trying to help you learn something and get some work done that's useful. And so if the way we're organizing it is unhelpful, feel free to say it's unhelpful. I mean, one difference with the Zoom compared to the old in person is that we don't have a captive audience. So we have no way of keeping you here if you don't want to be here. So I guess the one question is if people would like to combine any of the projects together, or do you want to start with the list of projects that we have? We could work on it together. I mean, we can work on it till 3, and then if a group feels that it doesn't have enough people or doesn't have critical mass or is too big, that's why we had a 3 o'clock check-in to basically say, is this working out? Do we want to reorganize? And so on. Bobby, do you have any suggestions at this point?
Yeah, I think combining those groups into the same breakout room makes a lot of sense. Even if they keep working on their each on their own project.
So putting you Josh together
with the EMT.
Okay. All right. So let's see. How am I going to do this? I have not actually thought about how to. One problem for me with the shared poll results is it doesn't actually tell me who voted which way. So it makes it a little harder for me to assign people because I can't see the assignments.
Oh, are we able to make the rooms and let them go themselves? I'm actually not.
Yeah, so I can so what I can do here is I can create the rooms, and then you can self-organize. So for example, if Josh would like to join the thymus group to begin with, that would be fine. The one thing is then we need to make sure that there are no orphans. So why don't we do that? The other problem with Zoom breakout rooms is that as soon as you're assigned to a breakout room, you disappear from the main meeting. And communication, I can't chat with you back and forth to the breakout room. So once you're in the breakout rooms, you have to use the Slack or email to communicate. You can come out of the breakout room, but the communication's a little bit, we'll figure it out. But okay. Any other questions before we do this? I had a little bit self-sort first on the chat. That's not a bad idea. If people, do people have the list of, let me screen share the list of topics that were brought up. And then you can look at that list of topics and the people involved. And if you want to text or chat privately with any of these people, can you see that all right? Let me zoom that a little bit more. I'll give you 5 minutes to do a little bit of private chat to organize. And then I will open up the breakout rooms. And if there's a breakout room which has one person in it, I will try to fix that. And I appreciate your patience in organization. I think we can get started pretty soon. I mean, in a sense, it's difficult because there's so many good projects. I think I'd love to see all of these things. Well, okay, I guess I would suggest maybe we should, to begin with, combine the thymus and mucosal immunology groups, since they're both considering. Okay, I will do that. And so then we could say, instead of saying just thymus model, we could say more, and immune response model. And okay. And then maybe it makes sense. I don't know how people feel. Maybe it makes sense, at least to begin with, to combine the tube remodeling, the epithelial mesenchymal transition modeling, and the
Lateral line.
Lateral line, or at least combine the lateral line and the epithelial of the mesenchymal transition modeling, since those are very similar models to begin with. The tube remodeling is a little bit different because it involves links rather than convergent extension more than. Okay, AJ. I'm not sure, since it wasn't all delta notch, cross inhibitory signaling. Okay. Evan says, "Yep." I think Evan. I think the convergent extension is a great. I'm going to remove that epithelial mesenchymal one for the moment. Again, deleting it doesn't mean that you can't reform it or that you can't work on it. It was just trying to organize groups that are a little bit more substantial. So in that case, we would have this nice tissue engineering project, a project thinking about thymus modeling and other kind of immune responses, vascular tumor group, a bacterial motility group, a model on collective migration, maybe in lateral line or in tubulogenesis or in, well, actually, I've kept the tubulogenesis out as a separate one for the moment. But focusing perhaps to begin with on delta notch type signaling or other kind of cross inhibitory signals, microRNA and others. Tube remodeling and convergent extension so far is kept separate. If you go in and there aren't enough people, let us know and we'll reassign. And then the ovarian development, which I think is also a great project. Anybody unassigned at the moment who needs help? I have this heading, which is general issues, and we can go into that. Or once people have broken out, the other thing to do is to have the people just stay in the main chat and talk about it. Everybody ready for? Actually, before I open it up, let me just go through quickly something which people have already done to a large extent, but I wanted to talk about for people who haven't done modeling much before, which is there's always a temptation when you start building models that you want to start. Can people see it should say worksheet for developing a mechanistic biological model? I want to make sure everybody can see that.
Yeah, we can see it.
Okay, fine. Thank you. So there's always a temptation to start coding right away. And many people have already thought these problems through in very great detail. But I think it really is important, especially in a relatively short project like this, to think carefully about what it is you want to achieve over the next 3 days, so that at the end of the 3 days, you have something that's limited, but nevertheless useful done. So, for example, in the case of the lateral line, there were these 3 separate areas, all of which are really interesting. One of which is the rosette formation, one of which was collective migration, one of which was contact inhibited signaling. And so it might be useful to try to break those down, especially in the more complex models, to break them down to pieces that are more manageable. The other thing that I encourage people to do, and again, a lot of people have done this already, so for you, this is a repetition, but if you haven't done this before, is that I would encourage you to start by thinking carefully about the biological problems that you're trying to work on. And we have built this little worksheet, which can help people perhaps, which is to, and we've heard this in detail from many of the presentations, we start with the biological observations that you're trying to present. And we've heard a bunch of phenomena that people are interested in modeling. And we've also heard quite a bit about, in many of these cases, what kind of data exist in the literature. It's okay if a problem is pretty abstract. For example, convergent extension could be quite an abstract problem. It doesn't have to be tied to, say, convergent extension in frogs during gastrulation. And so it's fine to have problems that are fairly generalizable, but it's important to think about what kinds of data you could have to build the model, to validate it. And then trying to define as closely as possible what the hypotheses you want to test are, how you'll determine whether your model worked. It may be an eye test, namely, does it look like what I want? But it might be more than that. And then begin to try to define what the components, physical components, what kinds of cells, what kinds of ECM, and so on, and what the key biological processes that are involved are. Is this random motility, cell adhesion, contact signaling, and so on. And I would drill down a little bit more and try to think about the flow of information in your system. We've seen a lot about how signals are transduced in the various examples that we have. Thank you, Josh. We'll look forward to seeing you later. The kind of signals that are transduced and how they're transduced. Is there contact signaling? Is there mechanical signaling? Is there other kind of signaling involved? And where the signaling occur? What length and time scales are involved? So that importantly, is it reasonable to build a model of this kind? Role of noise. And then ideally, I would try to write down a diagram of what you believe is going on. Biologists classically use diagrams to explain their understanding. And I find diagrams are extremely helpful. They form the scaffold on which you'll build all of the mathematical and computational models later on. The more detailed that qualitative description of what's going on is, the more likely you are to be successful in building a model. Once you have that model framework, then you have to think about how to make it mathematically intelligible. Ideally, you do a little bit of research on existing models, and people have shown some already. Discuss what possible gaps there are to fill in, and then what you can use. So we've heard some examples where people had ODE models of particular phenomena that they might want to spatialize. That's a very helpful thing to do. And then eventually, once you've done that, we have to start thinking about building a simulation. And we can come back to the stages of that simulation. But the more you can map out the biology clearly to identify what things are you're going to include and what you're going to leave out, I think the more likely this is to work. And so I don't want to take too much time. As I say, everyone who did a presentation had already thought through these issues pretty carefully. But for people who haven't done this before, I wanted to give this worksheet. Still worth, I think, filling it out for your problem of interest. And there's a preprint on the website in that Google Drive, which walks through the process of building model definitions in much more detail. Okay.